Hello friends and hello students. Welcome to the third lecture in the series of microbial cell culture and its applications. So, in the last lectures, lecture 1 and lecture 2, we have studied that why biotechnology is important, what is required for the microorganisms to grow. So, carbon source, nitrogen source, what kind of sources they are at laboratory scale and the scale up. Then once you have those sources including trace elements, cofactors, your antifoaming agents, water. Once you have this, how you go about in doing the cell cultures, and there we studied basically what are the ways to do cell cultures, its requirement of sterilization and mixing, what are the equipments that are required for doing sterilization as well as mixing, and basically running the cell cultures. So, essentially shake flask, baffled flask, bioreactors, especially we focused on stirred tank reactor, and there are other kinds of bioreactor as well. After that, we had a look at cell growth, that essentially how the what kind of cell growth happens, like it is a what kind of not really cell growth, but like what kind of the reactor is in, like whether it is a batch reactor or batch phase, batch system, fed batch system or a continuous system. So, we had a look at all this things, and now we will look at essentially the measurement and kinetics of microbial growth, okay, in this lecture. Why do we need measurement? First, why do we study kinetics? So, these are quite an important questions. So, for measurement, so if you do not measure the microbial growth, then basically you have no idea what is going in your system. Okay. So, to understand your system, you really need to know what is going on inside the shake flask or a reactor, and for that you need tools. Okay. And once you understand the microbial growth, it has a direct impact on the production of metabolites. So, if basically your microbial growth is affected, then you can have a look, then you know the reasons why your metabolite production is low or high or different or more contamination and many other things that can go in that. So, essentially you really need to understand how is your microbial growth is, and basically you can use the microbial growth and the data from that to understand the biological process, and this understanding will help you for proper utilization of your substrate and maximizing and optimizing the production of metabolites. Okay. So, that is why it is important to understand the measurement of microbial growth. Why kinetics? Okay. Kinetics is interesting because as we have discussed in the previous lectures, that time is also a factor. A product that is formed in 12 hours is much more better than a same product formed in 24 hours, because you lose time, and the time is directly related to the cost of the production. Okay. Now, I will give you a very simple example. In the auto industry, so car manufacturers, trucks manufacturers, they have moved away from managing an inventory of all the parts that they need. What they do is, there is concept called as in time inventory or on time inventory. That means that if the inventory is required today or tomorrow, then the parts would come in couple of days. So, if for example, any stoppage in that inventory chain is, so if any 5 minutes, 10 minutes, 20 minutes differences that might come because of the border check or because of more bureaucratic processes, then what it will lead is that every 5 or 10 minutes would cost somewhere at the range of $100,000. Yeah. So, you understand the value of time in the industrial processes. Another example of importance of time is, you buy a machine for, let us say, 10 crore rupees. Okay. Now, this machine, if this machine is continuously working, and these machines are designed to work continuously, 3 shifts, 8 hours, 8 hours, 8 hours. If these machines are working continuously for 24 hours, then you are producing enough output, and you have actually calculated the size of that machine based on the output required, and assuming that you are going to work 24 hours a day, and there would be some downtime, maybe 5%, 10%. So, that is already calculated in. But if the process takes much more time than what you have envisioned, then basically your machine, a 10 crore machine, is giving half the output in same amount of time. That means your production is gone half, right, and then basically this leads to large losses in the large losses in the production, okay. Or the other way would be, it increase the cost of the production. That means the product that is you are producing will not be cheap, but its cost has increased, okay. That is why kinetics is important to understand from industrial point of view. Of course, from scientific point of view also, kinetics can tell you a lot of different things, that how microorganisms are going, what is their behavior, what kind of things they like, how we can optimize this the data that is coming from the kinetic data that is coming, how we can optimize it to essentially use for larger production or better production, okay. So, that is why both measurement and kinetics is important. So, how do we measure the microbial growth? Okay. But before that, how to measure, okay, we also need to know what to measure. Okay. So, what essentially is microbial growth? Okay. We can say microbial growth is essentially increase in the population of microorganisms. This is the most simple definition. Okay. As the microorganisms increases, their population increases. So, number of microorganisms increases, also number of internal chemical component like DNA, RNA, metabolites, protein concentrations, all of this thing increases, okay. And this increase is what you can say as microbial growth, okay. Different kind of microorganisms grow differently, okay. In bacteria, the growth is by binary fission. In yeast, it is a budding process. In fungi, it is a chain elongation and branching. In viruses, it is intracellular duplication in the host cells, and it is completely different process. But for example, let us look at how the binary fission in bacteria happens. Okay. So, binary fission in bacteria is actually quite straightforward. You have a bacterial cell, okay, and then you would have a division over here. So, next step would be somewhere something like this, still attached, okay. Everything is divided, it is divided over here, and then basically you will have two bacteria that will be produced, okay. So, that is how binary fission happens. In the case of yeast, so in the case of yeast, it is budding process, okay. So, what happens here is that you have a yeast cell, you have a nucleus, this basically goes, it becomes, let us say, a bud is formed and this nucleus is elongated, okay. Then basically what will happen is that you will have here and you will have a daughter cell over here, and then eventually it becomes like this, bigger, okay. So, this is a budding process in yeast. In fungi, this is an chain elongation and branching process, okay. So, here basically what happens is that you have spores, and then the microorganisms, the fungi is formed, and then the chain is elongated, and then the branching would happens and things like this. One important thing to note here is essentially yeast and mold are types of fungi actually, okay. Mold we have not used, but you would have seen it in the houses. Basically, is a type of fungi that grows into a multicellular filaments, okay, while yeast is a type of fungi that grows as a single cell, okay. So, the budding process which is there for yeast multiplication, it is actually a fungal multiplication method as well, okay. So, this is just to clarify that I have put yeast and young fungi in the slides as two, but that was there was a reason for that, but yeast is actually a subspecies of fungi. Viruses are very interesting. I would encourage you to look it in the Wikipedia or the internet, that how essentially viruses are propagated. They actually are called as essentially the line between life and death. So, viruses on their own cannot survive, they need a host, and this host can be, as you now very well know, this host can be even humans. And also there are some viruses that affect only bacteria, and this viruses goes inside the bacteria, get themselves integrated into the DNA of the cell, and then use all the machinery of the host cells to increase their numbers, and then at the end of the day they burst out and do the cell lysing. In some cases they do, in some cases they do not do. So, there are many different kinds of things. So, it is actually a very interesting topic to have a look at it. Okay. So, this is how the microorganisms grow. Once we know microorganisms grow, what can we use this thing to essentially learn something. Okay. The growth of the microorganisms, we can use it for basically measurement of the microbial growth. Okay. So, let me clarify this statement. The microorganisms which are growing, so their population is increasing, okay, and this basically we can use it for the measurement of microbial growth. So, as a parameter, the population increase as a parameter to know about the microbial growth, fine, and then we can also study their growth kinetics and specific growth rate. Okay. So, there are two topics, two important points over here in measurement and kinetics of microbial growth. One is the measurement of the microbial growth. So, how do you measure it? And basically what you do is you can measure population, but you can also measure biomass. And in the second is growth kinetics and specific growth rate. These are the two things that basically that you can actually are required for seeing how the culture is growing and things like that. So, first let us look at the measurement of microbial growth. So, what is microbial growth? As I talked earlier as well, it is basically orderly increase in the components in the presence of suitable medium and the environment. Okay. So, a conducive environment for the growth, the substrate and everything is provided, and the microorganisms population increases. Okay. How can we measure the microbial growth? This was what I was talking about earlier. You can measure it by either measuring the total cell number that has increased, or you can measure by cell biomass, that how much is the mass that has increased. Okay. Then there is another question, and this is very important question: how to compare microbial growth under different situation. Okay. So, basically we want to compare. So, one important thing is that why do we even want to compare? So, the point is that two microorganisms grow at a different rate in different situation or even in the same situation. Okay. In some cases same microorganism would grow at a different rate in different situation. Okay. So, let me clarify this statement little bit more. We have an microorganisms x1 and we have an x2. Okay. x1 in 10 minutes become x1 plus, in 10 minutes it becomes x1 plus x1. So, 2x1. In another 10 minutes it would become x1 plus x1 plus x1 plus x1. Okay. While x2 is a different microorganisms and they are under the exactly same conditions. Okay. So, x2 require 30 minutes, okay, and it becomes x2 plus x2. It requires another 30 minutes and it will become x2 plus x2 plus x2 plus x2. Okay. How do you basically say x1? So, we know that time is a factor and we know that shorter time is better, but how do we say basically x1 is better than x2? Okay. If you are just growing it, you will not know. You come up with this kind of time. This is a characteristic time. It is called as doubling time or generation time. Okay. So, what this is called as? This is called as T D doubling time. It depends on the process, but typically you like to have a shorter doubling time. So, in this case, under the same condition producing same product, okay, we know x 1 is better, right, x 2 is not good. Okay. Doubling time is also important in case you have same microorganism. Okay. It is not as if that only one it is used to differentiate between two microorganisms. It is also good if even if you have one microorganism. Let us say you have an x 1 and x 1, but what you have is different conditions. What does this different condition means? These different conditions mean different. Okay. What this different can be? Substrate, temperature, pH, right, cofactors. So, many things. Now, if we run this at different time and we use the same thing, 10 minutes x 1 plus x 1, another 10 minutes x 1 plus x 1 plus x 1 plus x 1, and here x 1 30 minutes x 1 plus x 1, another 30 minutes, this will come out to be x 1 plus x 1 plus x 1 plus x 1. Okay. So, this thing again we know that x 1 in condition, let us say cond 1 is better than x 1 in cond 2. Condition 1 is better for x 1's growth and product formation compared to x 1 in condition 2. Okay. So, that is how we can actually compare, and the time is the doubling time is a characteristic parameter. Okay. Now, let us come to another area which is which we are discussing, that we know now that the doubling time is good, is a good characteristic. We also know we want to measure microbial growth. Okay. We also want to measure microbial kinetics. These are important to understand the process and everything, but how shall we do it? Okay. What are the ways to do it? So, the ways to do it are two major ways that are used very frequently are first measuring the dry weight of the cell material. Okay. So, for instance 1 billion E. coli cells weigh around 150 milligram. So, every cell would on an average would weigh more or less same. So, with time the weight, the dry weight of the cell material should increase. That is the first method. The second method is that absorbance of cell suspension in a spectrophotometer. As the cell concentration increases, their absorbance would increase, and based on that basically you can back calculate the cell concentrations. Okay. Other methods are wet cell measurements, turbidity measurements, ATP measurements, viable plate count and Coulter counter. So, let me come to the other methods first, and then I will explain how to do the measurement dry cell weight method as well as the absorbance method. Okay. So, turbidity measurement is very clear, you measure the turbidity. There are instruments that are available to actually measure the turbidity, and they would just say how turbid is basically your system is or your solution is, and based on that basically you can actually back calculate the cell concentrations. Okay. So, it is still not a direct method to calculate the cell concentrations. ATP measurements, what is ATP? ATP is adenosine triphosphate. This is actually the currency of energy in the cells. this is useful in driving all of the cellular processes. Okay. So, if you measure the ATP, the increase in the number of ATP correlate with the increase in the number of cells. Okay. So, that is how you can actually measure, again indirectly measure the number of cells. Okay. Another method is very useful in the laboratory is the viable plate count. So, you basically plate the culture on the plates and then measure and count the colonies that have grown, and each microorganism would grow one colony, and that will be called as CFU, colony forming units, and based on that you can actually back calculate the number of the cells. Okay. Other method is Coulter counter method, okay, and that actually measures the impedance. So, maybe it makes sense to actually let us look at first a plate count method, that is an interesting method, and then we can go to other methods. Okay. So, in the plate count method, basically what you do is you have a cell suspension. Okay. This is your cell suspension. Okay. The red ones are your cells. Okay. So, what you do is you take small amount of this, maybe 10 microliter or 30 microliter. So, I will maybe write it bigger. You take 10 microliter or 30 microliter of it, you put it on a plate. This plate, what does this plate? This is a agar plate. So, agar is a medium, okay, and you can put other nutrient as well, and it is solid, but not rock solid like a jelly. Okay. And you basically have agar with medium, which and it has all the requirement for the microorganism to grow, and then what you do is you add 30 microliter on the surface of it. Okay. So, you have a plate, the 30 microliter goes on the top of it, and then basically you spread it. So, with a spreader, basically how you wipe the floor of basically how you wipe the floor with a wiper, or how the wipers wipe the wind screens of the car. That is how you basically distribute it, and what will happen is that after that you incubate. This incubate at temperature and just keep it there, and then what you will see is something like this, specks. So, this is not one microorganism. If you can see it with naked eye, it will be basically circular. So, I just put across just to make sure you understand, but it will be kind of circular shape, okay, and separate. Okay. This is not one microorganisms, right. Basically, if it is one microorganism, then essentially this is very big, you cannot see it with eyes. What has happened is that we have taken 30 microliter, very small amount, and we have plated on it, and each of this is called as CFU, which is colony forming units. Okay. So, initially each one of them started with one microorganism, and then it divided and formed the colony. Okay. And each separate is one microorganism. Okay. And you know that you have put 30 microliter here. So, you can back calculate, if in 30 microliter you have 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8. If you have 8 microorganisms in 30 microliter, how many microorganisms you have in 1 liter? So, this is a straightforward calculation. So, that is how basically you will calculate it. Of course, this does not happen like this. You need to, in some plate you add 30 microliter, in another plate you add 10 microliter, in another plate you add 5 microliter, in another plate you dilute it and add 10 times diluted 10 microliter. Okay. So, that you can distinguish between the colony forming unit, because it can also happen that your colony forming unit looks like this. Okay. So, here you cannot say whether it is 1, 2, 3 or in this is 1, 2, 3, 4 or okay. You understand what I am saying? So, in that case this is not a good dilution, you need to dilute more. So, this is how basically you will measure the microbial growth using colony forming units or plate count method. Okay. Another method that is also interesting is Coulter counter method. This is also quite interesting. What they do is, again it is an indirect method. What they have is a circuit here, okay, and essentially they measure impedance. What they measure? They measure impedance. Okay. So, when there is nothing, it is a liquid here, okay, media. So, they know the impedance of the liquid, and it is so small that only one cell can pass at a time. Okay. So, when the cell passes through it, basically what happens is that there is a change in the impedance, and once there is a change in the impedance, basically they know that the cell has passed, and once they count it as one cell, and as the number of cells passes by, it can continue to count. So, for instance, it can count 10,000s of cell in few seconds. So, it is actually a very good method for a very quick determination. Okay. So, that is your Coulter counter method, but the method that is most used in the laboratories in biotechnology is dry cell weight and absorbance. So, how does the dry cell weight method works? Well, dry cell weight method works like the following. You have a reactor and it is full like this. Okay. The volume is, let us say, 1 liter. You take 10 milliliter of it. So, this 10 milliliter would have the biomass in it, and you make sure that it is homogeneous. So, you should not take the 10 liter which is deposited over here. Okay. So, if you take 10 milliliter and you basically dry the volume on a plate, crucible. So, when I say drying, what does it mean? We just want to evaporate the water. So, you can dry it at 105 degrees for 12 hours. Okay. Now, what you have done is this 10 ml, what you have done, this 10 ml that you have taken is you have put it in crucible. So, this is crucible. Let us say, and you have measured the weight of empty crucible. So, weight of empty crucible is C empty. Okay. And after 24, and you basically dry it at 105 degrees 12 hours, and now you say is C dry. Okay. So, C dry minus C empty will give you dry weight of cells. Okay. This dry weight of cell is for only 10 milliliter. Okay. And your reactor is 1 liter. So, you can back calculate that how much amount of biomass is in your 1 liter of reactor. So, this is how you do dry cell weight. What is the limitation over here? Can you think of a limitation? One of the limitation here is you require at least larger volumes. Okay. You cannot do it with 2 ml or 1 ml, because your measurement C dry, C empty, there would not be that much difference that the machine can actually, your weighing machine can differentiate that difference. So, one of the challenge is the volume that is required here. To overcome that, there is another method that is actually quite nice, that is your absorbance of cell suspension in a spectrophotometer. Okay. This method is actually based on Beer-Lambert's law. Okay. What this law says is essentially, if your light is passing through this section, okay, and let us say this is L. Okay. So, and if this is filled with something, your light will be scattered and the amount of light that is absorbed would be different. So, normally you can say A adsorbent transmitted is equal to A absorbance epsilon and L. Okay. It is called as Beer-Lambert law. Okay. And what happens here is that if, for example, I put a crucible, it is called as not crucible, but cuvettes. I have a cuvette, I fill it here, it normally require only 2 milliliters, okay, and I put this and A incident. So, this is A I, let me write it like this, A I, and this is A transmittance. Okay. Of course, A transmittant would be smaller than A incident because it is adsorbed, right. Okay. And how much it is adsorbed, which would be basically your A incident minus A transmittant, is proportional to your cell concentration inside the 2 milliliter. Okay. Here again, based on this, basically if you have a standard curve. So, for example, if you know concentration of cells and absorbed, okay, if you know that this is curve and this equation is y is equal to mx plus c. Okay. So, now when you do an experiment, you take a cuvette, you fill it with 2 milliliter, you put this in your spectrophotometer. So, this is a machine that measures the absorbance. Okay. So, you put it over here and then you get an absorbance value. Okay. Let's call it as Z. So, basically what you have, you already know Y, you already know M, X and C. Okay. You basically know this equation y is equal to mx plus c. What is here absorbance? Absorbance is your y here. So, basically you will just say Z is equal to mx plus c. You know the c, you know the m, you can calculate x. Okay. And this x is your concentration of cells. Okay. It is not dependent on if you fill it here or if you fill it the cuvette here. So, if you fill the cuvette till here or you fill the cuvette till here, it is not dependent on that, because the length that I talked about is this, light is passing through this, not this. Okay. If you see here, there is one interesting observation, and that is that in this method there is a requirement that the cells do not settle down, right. The cells should be in suspension. You know why? Very simple. If the cells are this, this is a suspension, this is a homogeneous suspension as we discussed in the last class, this is light and this is detector of spectrophotometer. Okay. Now, imagine that your cells are settling down. So, how will it look like? If the cells are settling down, then should be green in color. So, the green colors are our cells. This is incident light. Okay. This is your detector. Okay. So, this concentration, it will only measure this cells, while this would measure this cell, while there is this concentration here is smaller than this concentration here. Okay. So, essentially you are giving lower concentration than what you have in the system. Okay. And that is why it is important for absorbance of cell suspension in spectrophotometer is that the cells are still in suspension and there is no visible settling. So, how do you ensure it? Well, you just add and you measure. So, you do not wait for 20 minutes, 30 minutes to measure, you just as soon as you add you measure, and in that case basically cell would still be in suspension, they will not settle down that quickly. Okay. So, that is how basically we can measure the microbial growth. Then as we have talked about growth kinetics and specific growth rate, so we want to know basically that how do we talk about this and what is like what are the importance of that. So, essentially growth kinetics and specific growth rate, what they will give it to you? They will give the doubling time to you. Okay. And that will be very important for us to design a system to compare the microorganism and to see how our system is performing. Okay. So, in growth kinetics and specific growth rate, the characteristic of kinetics can be defined by generation time or doubling time, and this is how basically I just explained in this lecture before. I will just quickly explain it again. What is this? Is you have x1 microorganisms in 30 minutes, it becomes x1 plus x1. So, 2 times x1, another 30 minutes it becomes x1 plus x1 plus x1 plus x1, this is 4x1. TD, the doubling time is 30 minutes. So, this is your generation time or doubling time. Okay. Bacterial growth phase, so the doubling time is related only to your log phase or exponential phase. Okay. So, what is that? That is essentially this thing. This is time and this is biomass or concentration. So, you know that the growth phases. So, you have an lag phase, then you have an exponential phase, then you have an stationary phase, and then you have a death phase. Okay. So, this is your lag phase. This is exponential phase or also called as log phase. This is stationary phase and this is death phase. Okay. You remember what was in the lag phase? Well, in the lag phase the microorganisms is not utilizing a substrate too much, but what it is doing is essentially producing lot of enzymes and preparing itself to utilize the substrate. Okay. So, it is kind of a war preparation. Once that preparation finishes, it goes into the log phase where it starts to divide and its growth rate increases. Okay. Once that happens, then basically there is a stationary phase where the substrate is utilized. So, it is not growing anymore, it is just maintaining its population, the number of cells dying are number of cells produced, and once it passes through, then the cell death starts to happen and the microorganisms start to die, the population starts to decrease. Okay. One point is that is the growth rate of the cell constant? Specific growth rate is constant, but the growth rate of the cell is not constant, because as the number of biomass increases, the growth rate increases. Okay. So, when we are talking about generation time or doubling time, we are talking only about the log phase, not about the lag phase, neither about the stationary phase nor about the death phase, but only about the log phase. Okay. And how do we calculate doubling time? So, there are two ways to do it. In this lecture, we will only look at how to do it with biomass. In the next lecture, we will look at how to do it with cell number. So, to measure doubling time, we can either model it based on the biomass, which as I repeated and as I just told, we will do it in this class, while for cell number, we will do it in the lecture number 4. Okay. So, then how do you do it? That is a question that we want to answer. So, let us look at this. What is X? X is our biomass concentration. What is time? Time is T is time. Time is basically time of incubation from the starting point. Okay. And what is mu? Mu is our specific growth rate. How this has come into picture? So, let me explain this. Again we take that example x1 becomes x1 plus x2, x1 plus x1, and then again x1 plus x1 plus x1 plus x1. So, 4 x1 2 x1, okay, and this is your doubling time. So, is it okay to say that the rate of change of x with time is proportional to x? That means dx by dt is proportional to x, because see here, it is started, the time is the same, this is t doubling here, t doubling here. It started with 1 x, the biomass increased twice, but biomass increased twice, but the total biomass increased 4 times. Okay. So, the rate of change of x with time is dependent on x, that is your first order kinetics, okay, and you can write as dx by dt is equal to mu x. Mu is the constant of proportionality or specific growth rate. Okay. So, if dx by dt is equal to mu x, okay, is this okay sign, or should it be dx by dt is equal to minus mu x? This is right, this is not correct. Why? Because the sign is should be positive, because the larger the biomass, the larger is the growth rate. Okay. So, when dx by dt is this, if you integrate this, okay, you have to transfer x here. So, or it is a simple, let us, if we do a simple integration, it will come out to be x t equal to x 0 e to the power mu t. Okay. We do a definite integration. What is x 0 is initial biomass concentration. Okay. What is x t? X t is biomass concentration at time t. Okay. And what is mu? Well, mu is specific growth rate. Okay. So, what we have? We have x t equal to x 0 e to the power mu t. Okay. Now, if I take ln of this, okay, which means ln of x t is equal to ln x 0 plus mu t. Okay. So, ln of x t minus ln of x 0 divided by t is equal to mu. But if you look at this equation and if I plot time, okay, and I will just write it like this, ln of x, okay, then the slope of this line would be equal to mu, right. So, in an experiment, so how does it matter? It matters because in an experiment I can measure x and I can measure time. I cannot measure mu directly, I can measure x, as we have seen, either by your dry weight method, spectrophotometer, right. I can measure x, and time of course I can measure by simple watch. So, it can be like this 0 0, let us say time in 5 minutes is 0.2, time in 10 minutes is 0.25, and so on and so forth, and once I plot ln x and mu, I can get the slope, and I would know x 0 because x 0 is the starting point, okay. Or and let us say x 0 would be what, at time I can put at time t is equal to 0, I do not put x 0 as 0, but I can put x 0 as very small 0.01 like this, and then I can get the value of mu, okay. Now comes the other part where we are looking at the doubling time, okay. So, x t is equal to x 0 e to the power mu t, okay. So, what is doubling time? Doubling time would be t d, where basically x 0 become 2 x 0. Now, that means at t d x t is 2 x 0. So, let me just replace it over here, 2 x 0 equal to x 0 e to the power mu t d, okay. That means I can cancel this, okay, and then I have 2 equal to e to the power mu t d, okay. So, what I have? I have 2 equal to e to the power mu t d, okay. What is t d here? T d here is doubling time, okay. What is mu? Mu is specific growth rate that you can either find by equation or you can find by the slope of semi-log plot. No, why semi-log? Because you take x axis as time, which is normal, and then log of x t, log x t minus log x 0. Okay. Now, so mu is a specific growth rate. So, now if I take natural logarithm, which is ln, so ln 2 is equal to mu t d, ln 2 is equal to 0.693. Okay. So, my doubling time is equal to 0.693 divided by mu. So, I can calculate my T d by dividing 0.693 divided by mu. How can I calculate mu? I have already shown you. You can plot. So, how to calculate mu. You can plot ln x and time, okay, because you have an equation ln x t is equal to ln x 0 plus mu t, okay. And this x, as I have said, you can calculate or measure by biomass, or you can use directly this equation at any point ln x t minus ln x 0 divided by mu is equal to divided by t is equal to mu, okay. And this t is the time for the incubation where the biomass concentration is x t, okay. So, essentially this gives us how to identify mu and doubling time. It is important to note, as I have already said, mu changes during the different phases of batch growth. So, the doubling time that we have seen is only for the log phase or the exponential phase. Also note mu is your specific growth rate. So, that it means it is affected by the temperature. So, growth temperature means the temperature that you are using in the incubation, medium composition and other environmental factors like pH, your pH or the cofactors that you have added or not added, okay. So, this is how basically we can talk about the growth kinetics and a specific growth rate and can find out the doubling time. The other method to do it is based on the numbers of the biomass, numbers of the microorganism, and this we will do it in lecture 4. So, in lecture 4 we will look at this part as well as the scale up, that how you do the scale up. Thank you very much and have a good day.